Hi, everyone. So today we're going to be discussing the insight into the mechanism of the Influenza A Proton Channel. This study was conducted by Mukesh Sharma and his team at Florida State University's Department of Biochemistry. And it draws on a lot of previous studies regarding the M2 proton channel to investigate the structure of its functional residues and propose a mechanism for how protons get across the channel. So M2 is the star player here. It's a transmembrane protein on the viral envelope, and it's active acid activated and necessary for viral replication. The mechanism proposed in this study relies on a cluster of residues believed to be responsible for conductance. Proton conductance is a very fancy way of saying protons moving from outside of the viral envelope to inside of the viral envelope. And this sequence of residues has been responsive to antiviral drugs, and improved drugs are necessary for strains of influenza A that have been mutated and become resistant to the current antiviral drugs. Unfortunately, I'm sure we can all consider why having this antiviral drug be effective, even against mutations, is really important. We can even see in the COVID-19 pandemic how mutations in the COVID genome have led to less effective vaccinations. And this is just like another instance where staying proactive against these mutations is really important. So I wanted to start with a short background on the steps towards viral replication for influenza A. This isn't all of them, just a brief view to situate us in this discussion. So the first step would be the virion entering the cell by endocytosis. And then the activation step would be when the M2 protein on the viral envelope is activated by the low pH of the lumen environment. And then protons can conduct across the virion membrane, lowering the internal pH, which will eventually lead to viral uncoating, which is crucial to allow like the viral machinery to enter the nucleus and begin transcription. This study is mostly narrowed on the second and third steps of this process, as we can see in the highlighted slides. As we mentioned earlier, activation and conductance are reliant on a specific stretch of residues in the M2 protein. In earlier studies of the M2 protein, the transmembrane residues, number 26 through 46, were thought to be the most important functionally. However, the authors cite a study by Ma et al. At Northwestern University's Department of Biochemistry that suggests a greater length of functionally important residues exist beyond the transmembrane domain. So by taking these extra residues in addition to the transmembrane domain, we get what is referred to as the conductance domain of the M2 protein. And that's what we're really zeroing in here. So a closer look at the work done by Chunlong Ma and his team at Northwestern was essentially researchers synthesizing M2 and specific fragments of M2. These specific fragments are the transmembrane domain and the conductance domain with frog oocytes. And then the specific activity of these fragments was tracked in comparison to the specific activity of the entire M2 protein. So the top image is the top of the image is a specific activity of the entire M2 protein. And then the one below it is the specific activity of the conductance domain, which we can see is like nearly identical. And then the specific activity of the M2 transmembrane domain is still pretty good conductance, however, not as comparable to the entire protein as the M2 conductance domain is. This is what informed the designation of the conductance domain for the study.
And moving on to structure and function, first, we wanted to discuss solid state NMR because it was a technique that was very important to this study in characterizing and describing the function of this specific protein. And so they use a specific NMR here called Pissema Nmr that's specific for membrane proteins, and it stands for polarization inversion plus spin exchange at the magic angle NMR. And basically, it creates a correlation between chemical shift and heteronuclear dipolar coupling. And this can be used to find out constraints for simulations of the molecule in computers. And that can help out to figure out how it functions and what its structure actually looks like. And this is a two-dimensional NMR technique, and the chemical shift is nitrogen isotope and isotropic chemical shift, and the dipolar coupling is between nitrogen and hydrogen. And some limitations of this technique is it's an estimation, and there's also difficulty with protons at the end of the protein because of proton exchange with the environment. That's more prevalent there. And something special about solid state NMR is it's fundamentally different from solution-based NMR because everything is more constrained since it's in a supported solid, whereas in a solution, everything can move around and be more dynamic. And this causes some parameters that would average out to become noticeable in solid state NMR, which is both to the detriment and advantage as we can characterize things better, but also we have to be more careful in how we treat our data. And here, the cutoff was determined as 125 ppm chemical shift, where less than that is part of the amphipathic helix, and above that is the transmembrane helix. And I'll introduce those concepts in the next slide. And the program that was used here to analyze the restraints was called PPath, and it was very necessary for this study. And here's an example of what the NMR actually looks like. And the A, B, and C are different N15 labeled amino acids, where the first one is valine, and the second one is leucine and phenylalanine, and the third one is several others. But what I want to draw your attention to here is the diagram D, which has the example of these patterns called Pisa wheels that occur in the Pissema Nmrs. And what these are basically a picture of what the helix looks like because of the properties of periodicity of the helices. The I plus fourth, and I plus seventh residues on these helices show up near each other in these circular patterns, which gives us an actual almost 3D image of what these molecules look like, which is very, very key in determining 3D structure using this NMR technique. And below we can see in diagrams E and F is checking results against refined structure in a program. And this just helps to make sure the data isn't completely wrong and make sure we're getting a realistic results. And so how does the structural determination actually occur after these techniques? So the first step is to determine the monomer structure with Pissema restraints in a program called Explore NIH. And here, the restrictions that were determined by NMR were plugged into the program as parameters, which allowed narrowing down of what the monomer might actually look like and behave like. And the second step was to scale this up to the tetramer, so 4 monomers, which was not very complicated, but where it gets more complex is to try and mimic the native environment where this protein is. So as you can see from the figure below, this concept of detergent micelles, which is basically a circular lipid, a circular structure made of lipids, was previously used a lot of times, especially in this type of research, in order to characterize lipid-bound proteins. And it's a very non-native environment, so it may or may not be actually representative of the protein. So in this study, in order to mimic as best possible the native environment, a liposome was used to mimic the bilayer. And of course, this can't 100% mimic it since the bilayer is so complex in reality. However, it gives a much more accurate model compared to that, compared to the detergent micelles used in the past. And the fourth step is to use quantum chemical techniques to narrow in on areas of interest. And it's unrealistic to do it for the whole molecule, but it's definitely a great idea for some parts of the molecule of the protein. And M2 is a tetrameric protein, so it's made out of 4 monomers, and each of these monomers contains 2 different helices. And the first one is the kinked transmembrane helix, which is residues 26 to 46, and it's kinked around glycine 34. So on the right, on the top on the right, you can see the red highlight in the middle of the blue helix, and that's the glycine 34 where the kink occurs. And if you look at the figure on the bottom left with the green glycine 34s, you can see what the kink actually looks like and how the molecule or the helix is literally bent over. And the second helix is the empathic helix, residues 48 to 58. And it's thought that the role is to interact with the lipid bilayer to act as an anchor or a base for the entire protein. And as you can see on the bottom left figure, those flat helices on the bottom that forms the L with the top helices are the amphipathic helix. And there was confirmed a tight rigid turn between these two due to the Pissema Nmr of leucine 46 and phenylalanine 47 being very different and belonging to characteristics of their respective helix, which showed that there was a sudden change. So there's most likely a very rapid turn. And here's just a picture of a Western blot that was done to show that M2, the molecular weight of M2 after lysing was 1/4 of the actual weight that we know it is, which shows that once it lysed, it became 4 different monomers that were each 1/4 of the weight of the molecule or of the protein. Moving on, what was very important here was analysis of the pore structure. So this pore structure is formed by the transmembrane helices, and it only contains polar residues, and it's basically a seal to stop protons from escaping where they're not supposed to. And tryptophan 41 on the N-terminus is known to act as a gate that can be opened or closed to control proton movement. And the study proposed that valine 27 on the C-terminus may be able to do that function, but it's not confirmed. And there is known to be an amantadine binding site, which is a antibiotic between, or an antiviral between valine 27 and histidine 37. However, this brings us back to the native protein technique. So in detergent micelles, you're actually not able to see this. However, when you're using a more native environment like the liposome, this reappears, which just shows us how important it is to mimic the native environment if we want to get an accurate picture of what the protein actually looks like. And below you can see a figure that just gives a little picture of where these are or where these are oriented in the protein. And moving on to how the monomers interact with each other. So the nonpolar residues link the monomers in the amphipathic helices. So these are very buried very deep in the molecule since they're between the monomers themselves. And this was actually proved with hydrogen deuterium exchange data to show that these parts of the molecules were of the protein were the slowest exchanging, which is consistent with what we think is the interaction there. And the approach of these monomers is actually facilitated by very small glycine 58 and is strengthened by interactions between phenylalanine and leucine. As we can see here, there's a couple interactions actually. And there's a specific sequence on the monomers, phenylalanine 47 and 48, that indicate lipid bilayer association, which is consistent with what we thought the function of some of the helices were. And there's also tethering at palmitic acid to serine 50 hydroxyl to hold to provide even more of an anchor for the molecule. And this protein actually follows the positive inside role where the cytoplasmic side stays mostly positive, as mostly positive residues. And this is thought to allow good interaction with some negatively charged lipids and allow further anchoring in the lipid bilayer.
All right, and now on to the function of how this works. So first off, this is what we're going to be looking at. So the hydrophobic pocket is an area that was once thought of as where Ramantadine, an antiviral drug that treats influenza A, binds in the transmembrane helix, which contains an aspartic acid hidden at the bottom. It's that D44 on that structure. And after our researchers determined the structure, they noticed that the helices in the transmembrane tilt and make the hydrophobic pocket wider, and the aspartic acid can't be accessed anymore. And this means that Ramantadine can't bind from the exterior. However, it had been previously observed as being binded because of a detergent environment, like Sinan just mentioned, that was used to previously characterize the structure. And that shows the importance of the environment that we look at our structures in. So in this structure over on this slide, on the left, we have here we have high pH, and at high pH, the transmembrane helices are packed tightly, and the tryptophan gate is locked through intermolecular interactions with aspartate 44, which is what we were just talking about. And at low pH, protonation of the histidine 37 imidazoles destabilizes transmembrane helix packing. So as we'll see later, this allows the hydration of the channel pore and proton conductance. The C-terminal base of the tetramer and N-terminal disulfide bonds keep the channel from completely disassembling. And this shows the HXXW quartet that we'll be exploring more now. So the place where the acid activation and proton conductance happens is the HXXW quartet, which is the histidine 37 to tryptophan 41 cluster. And the histidine 37 residues had pKa values around 8 and lower, meaning that the acid was relatively strong. At a pH of 7.5, the histidine was doubly protonated and shared between nearby histidines, which strengthened the hydrogen bond through resonance. And this histidine tetrad is a pair of imidazole and imidazolium dimers where the proton is shared. Here, however, it was found that the two imidazole rings in the backbone were parallel to each other, which is not as favorable as it being perpendicular. In the dimers, the N sigma 1 and N epsilon 2 atoms aren't part of the hydrogen bonds and are instead protonated and toward the C-terminal side. The N epsilon 2 protons interact with the indole in the tryptophan 41, so that's the other side of the quartet, while the N sigma 1 protons make hydrogen bonds with the oxygens of the carbonyls in the histidine, so in the backbone. So don't worry, I'm going to make this all make sense. This means that the histidine is essentially locked in the HXXW quartet, and the channel is blocked because the protons can't be released from the imidazole NH protons to the C-terminal pore. Also, the other imidazole nitrogens that can give protons are part of strong hydrogen bonds. So if the protons were exchanged from the N-terminal pore here, and there were some side chain rotations, the imidazole and imidazolium dimers would end up exchanging partners. And however, since the NMR data consistently showed a symmetric structure, on average, this partner exchange likely happens like super quickly on a sub-millisecond timescale. And if you look at this figure on the right over here, it shows the model for activation of the M2 ion channel, and it shows the transmembrane domain in the, that's like the green, and the red is the histidine 37, and the blue is the tryptophan 41. So it's also important to note that these are only for 2 of the 4 subunits. So on the top over here, it shows the scheme for a closed M2 channel. And the channel is closed when the pH of the outside is high because the histidine 37 is not charged, and the tryptophan 41 obstructs the pore near its cytoplasmic end. And on the bottom over here, it shows the M2 channel in the open state with a low pH on the outside. When the pH on the outside is low, the histidine 37 is charged, which allows for the rotation of the tryptophan 41 to a conformation parallel to the pores axis, which allows hydrogens to flow. And on the left, this figure here was created in PyMol to show all the HXXW quartets in the M2 protein. Those are highlighted red. All right, and then over here, it shows the structure of the HXXW quartet in the histidine lock state. So in figure A on the left, it shows the top view of the tetrameric cluster of 8 of the histidine 37 to tryptophan 41, the HXXW quartet, and it shows the histidine 37 as sticks and the tryptophan 41 as spheres. So here, each imidazole-imidazolium dimer forms a strong hydrogen bond between the N sigma 1 and N epsilon 2. And in each dimer, the other N epsilon 2 interacts with the indole of a tryptophan 41 residue through a cation pi interaction. And NMR showed us that the backbones have fourfold symmetry. On the right, in the other structure, it shows a side view of one of the two imidazole-imidazolium dimers. So this shows us the pieces we've just been talking about. So both the intra residue and sigma 1 HO hydrogen bond and the inter residue and epsilon 2 H and sigma 1 strong hydrogen bond can be seen. All right, and then so on the right over here, it shows a model for illustrating the opening of the M2 proton channel to hone in on a key point. So here, there's 4 stages of protonation that are shown. In A, so top left, all 4 histidines are neutral. And in B, there's 2 charges. In 3, there's 3 charges. And in D, there's a fully protonated histidine tetrad. So neutral is yellow and positively charged is green for the histidine residues. And the open state is shown with that like pink area. And the dashed lines all around in the histidine dimers show the strong hydrogen bonds. So basically, this shows that when you disrupt one of the dimer structures, it results in a high energy state where the system is willing to give up a proton and return to the paired dimer state. And this is supported because the second and third pKa's are separated by nearly 2 pH units. So it shows how difficult it is to disrupt the stable histidine locked conformation. And over on the left, it shows our M2 protein, and the blue is specifically just one HXXW quartet that we're looking at, while the red is other HXXW quartets in the same protein. All right, and then, so using a neutral pH to study the structure of the HXXW quartet, the researchers determined the mechanism of the acid activation and proton conductance. So they found that under acidic conditions in the exterior, a hydronium ion from the N-terminal pore attacks an imidazole-imidazolium dimer. This is now the activated state. And the now triply protonated histidine tetrad is stabilized by a hydrogen bond with water at the N sigma 1 site that there's now access to. And this is located on the N-terminal side. And another stabilizing factor is an additional cation pi interaction with a tryptophan 41 residue at the N epsilon 2 site on the C-terminal side. So using Ramon spectroscopy in the transmembrane domain with acidic conditions, this strong cation pi interaction with histidine 37 and tryptophan 41 was observed. And what Ramon spectroscopy is that it's a spectroscopic technique that's used to determine vibrational modes of molecules, and rotational and other low frequency modes of systems may also be observed. And Ramon spectroscopy is usually used in chemistry to provide a structural fingerprint by which molecules can be identified. So those strong cation pi interactions between histidine 37 and tryptophan 41 protect the protons on the C-terminal side from water access. However, when the helical backbone undergoes conformational changes, like a change in the helix kink in glycine 34 I was talked about earlier, in addition to like the movement of the tryptophan 41 side chain, it leads to the breaking of the cation pi interaction. This is now the conducting state, and the N epsilon 2 proton is exposed to water on the C-terminal side, which lets it be released to the C-terminal pore. When that proton is released, everything repeats. So basically, the histidine becomes locked again and is ready for proton uptake from the N-terminal. Then the proton releases to the C-terminal pore. And in each round of proton conductance, as the histidine changes from being locked, activated, and conducting for the HXXW quartet, the angle of the histidine 37 and tryptophan 41 side chains need to change, and they rotate. So these rotational angles, however, were discovered to be like much smaller than previously thought, and similarly, they're lower than what's needed for the imidazole-imidazolium dimers when they have to exchange partners. So in this figure right here on the right, it shows the entire mechanism. So it's the proposed mechanism of acid activation and proton conductance, illustrated with like half of the HXXW quartet from a side view. So on the top, it shows the histidine lock state with a hydronium ion waiting in the N-terminal pore. So acid activation is initiated with a proton transfer from the hydronium ion into the inner residue hydrogen bond between N sigma 1 and N epsilon 2. In the resulting activated state, the two imidazolium rings rotate so that the two nitrogens move toward the center of the pore. In addition, the protonated N sigma 1 forms a hydrogen bond with water in the N-terminal pore, while the protonated N epsilon 2 moves downward. So when the angle relaxes, to form a cation pi interaction with an indole, and that blocks water access from the C-terminal pore. The conducting state is obtained when this indole moves aside to expose the N epsilon 2 proton to water in the C-terminal pore. And once that N epsilon 2 proton is released to the C-terminal water, the HXXW quartet returns to the histidine lock state. And on the left, in red, the histidine 37 is highlighted. That's also a self-created image in PyMol.
So there are a few different studies that support the conclusions drawn from this experiment. First, the proposed mechanism suggests that acidification would only be possible from the extracellular pH and the N-terminal. And in a study where basic pH is applied to cells with M2, there's an observed decay of proton current, and this is consistent with the histidine locked state. So protons were not flowing into the viral envelope in these conditions, and it's thought that the tryptophan gate was responsible for blocking them. And in another study where tryptophan was mutated, proton flow was observed regardless of the pH. Similarly, copper was found to block M2, but only extracellularly, which supports that activation is only possible from the N-terminal side because it's thought that that copper would coordinate with histidine, causing this blockage. So M2 cannot be activated by other cations, which supports the proposed histidine tetrad. However, there is disagreement over how that tetrad is oriented, which we'll observe in a second. And also the pH that authors expected to begin activation and to saturate M2 was observed to be the pH that those actions happened in at other studies. So again, there is evidence that supports some of the conclusions made from the proposed mechanism. However, there are like a few critiques that are made about this study. One limitation of the methods used to analyze M2 was the strain on the flexibility of the protein's backbone, which would affect the structural analysis of the histidine tetrad. Other studies have proposed different orientations because of this lack of flexibility. And just one of the proposed orientations was a histidine box. Another thing that was inconsistent with other studies of M2 was the exact ways that protons enter the cell. There's disagreement over whether the protons enter one at a time through the proposed mechanism, or if there is some form of proton delocalization that occurs, allowing protons to enter the cell, or even a stream of water flowing protons into the cell. So that is all we have today. Thank you for listening to our discussion of this study, and have a good day.